Hello dear students. Today I'm going to explain about the some important objectives from the chapter sexual reproduction in the flowering plants, which are nothing but angiosperms. Right? Let's get started our video with the first objective, which combination correctly identifies the essential reproductive goals of a typical bisexual flower. We know that flower have a four whorls are there. In that outermost one is the which we called as calyx, which is the color one is the corolla. Next one is the pistil and stamen and pistil, right? So here they ask the correctly identified part, which combination correctly identifies the essential they asked. Essential means which are involved in the reproduction, right? Actually, these outer two parts, which are the calyx and corolla, are the accessory whorls, right? What are the main function for that? No, to attract the insects, which are the corolla function. Calyx is the give the protection to the innermost layers, right? So here option A, calyx and corolla they given. These are the non-essential organs, right? Are the parts. Second one, androecium and gynoecium. Androecium, gynoecium are the involved in the reproduction, right? That's the reason answer is the B. The explanation is the should remember easily CCPP from outside to inside. Double C, double P can use as a C square or P square. Outside to inside, the rule is there. Calyx, corolla. These two holes are the accessory, means additional, and androecium and gynoecium. Androecium for the male and gynoecium in the female are the essential gamete. They produce the structures. That's why answer is the androecium and gynoecium, right? Outermost one is the calyx. Second one is the corolla. Next one is the androecium. Last one is the gynoecium, right? The answer is the B. We'll go for the second one. If the innermost wall of layer microsporangium fails to develop. Okay, for example, here microsporangium, micro means what? Male. Macro means female. Okay, mega. We call as a mega using, right? If the innermost wall layer of a microsporangium, that's nothing but tapetum they have given, fails to develop. It's not developing, it's failed. What is the immediate biological consequences? It's a very good objective. See option A, the anther will fail to desist, means split open, they have given. Second is that the pollen grains will lack their productive sporospollen in exine. Exine is made up of right there, that was given. And C, the developing pollen grains will be deprived of critical nourishment. Last one is the meiotic division will be permanently halted, right? In this case, tapetum, we know that that's a function. Is the main function is the nourishment, right? That's the answer is the developing pollen grains will be deprived of critical nourishment. They lack of nourishment. Explanation is the while the outer three layers, you know that this is the microsporangium. It has a three layers. Outer is the epidermis and endothelium and middle layers are there. So these product the split the anther. The the tapetum consists of the dense multinucleate. This is a very important tapetum. What is the nature? Multinucleate cells dedicated entry the nourishment to the developing pollen. Now pollen has to be developed. No, it has a tapetum is very essential. This we can consider as a homologous to the human nerve cells are the sustentacular cells, also called as a well-known term is the Sertoli cells, right? They Sertoli cells giving the nourishment to the growing sperm cell. Here also for the pollen grains, these tapetum layer will be providing nourishment. That's answer is the 3. That is the third C is the option. Right? Next one. Yeah. During megasporogenesis. Okay, megasporogenesis regarding the female. A diploid megaspore mother cell, MMC, you are calling as MMC, diploid megaspore mother cell undergoes meiosis to form 4 haploid megaspores. In most angiosperms, what is the ultimate fate of the tetrad? Okay, tetrad, 4 there, no, undergoes the meiosis to form a 4 haploid megaspores. These 4, what will happen in the future? That's a question. Here, all 4 cells fuse to form the primary endosperm nucleus, right? Second is the 3 degenerate, leaving exactly one fundamental one, functional one, megaspore will be formed. Third is that they undergoes rapid mitosis to form a 16 free nuclei. To survive to form a synergids, other two will be polar nuclei. So here, see here, from the MMC, after meiosis, 4 haploid megaspores will be formed. 1, 2, 3. In that 4, 3 will be degenerate. 1 will be selected and promoted as a megaspore, which is the haploid. Because of diploid to haploid, here is the reductional division happened. The explanation is, see, this is known as the monosporic development. Monosporic means what? Mono means one, sporic means spore. One spore will develop. Rest among the 4, 3 will be degenerate. Monosporic development will be noticed. Only one megaspore, that's usually the Kalaza one, contributes to form a female gametophyte, while others will be degenerate. This is the female gametophyte will be formed. That's the embryo sac. Right? Correct answer is the second. 3 degenerate, rest of will be changed into megaspore. Right? Now see here. Question MCQ on the embryo sac. The mature female gametophyte is a botanical marvel of efficiency. Which statement accurately describes its final cellular architecture just prior to fertilization? Just prior to fertilization, what the condition in the complete, what you call female gametophyte? What it? In that, see here, antipodals are there and polar nuclei, egg, synergids. See here, calculate the how many cells are there? 1, 2, 3, 4, 5, 6, 7. Okay, total are the 7 cells. But how many nucleus there? Here 1. Polar nuclei, secondary nuclei there, no? Here 2. 2 plus 3, 5, 6, 7, 8. Okay, total 7 distinct cells housing the 8 haploid nuclei. That's the correct answer. See, it contains the 3 antipodals, 2 synergids in below, 1 egg cell is there. That's why 6 cells with 1 nucleus each. Plus 1 massive center cell. This is nothing but center cell. Containing the 2 polar nuclei. That's why I have to collect the 2 nuclei extra. Then that will be as a the complete Morwell efficiency of the particular female gametophyte is the 7 cells having the 8 haploid nuclei. Okay, that's the thing, interesting thing in the embryo sac. Yeah, this is the last objective for this today's video. Match the outbreeding device to its mechanism. They asking mechanism. Sunflowers, anthers maturing before its stigma. It is an example of dash. Okay. Here, see question, read the question properly. A sunflower anthers maturing before its stigma. Anther will be matured before stigma. Means what? Male is maturing before female. What it? That's called as the dichogamy. Whereas the hibiscus plant, if you take physically separating its anther and stigma. See here, this is the stigma anther. They physically separated, no? This condition called as hercogamy. Okay, dichogamy, hercogamy. Here is a protandry term, you know that. Dichogamy means what? No, different timings. The protandry means anther will be ripened first than the stigma. Okay, hercogamy means what? No, different positions, maybe height or the place. Okay, see hibiscus, we can see the hercogamy, right? Dichogamy we can see in the sunflower. Like that same thing we can see in the what do you call earthworm also. Earthworms are the hermaphrodite. even though there is no self fertilization, because of male and female they will be ripen, or they come into the sexual stage in the different timings. Like here also protandry means what, no, anther will be first ripen, release the pollen grains. At the time stigma won't be received. That's why it be depends on the cross pollination. Right, I hope you clear this one. Thank you.